Okay, good deal. All right, let me share the screen. Oh boy, now what happens? Okay, let me get rid of the emails. Get rid of that. And now, this is not where I want to be. Try to get somewhere close to where I was. Before y'all turn off the audio, I want to make sure we're on. Okay, this is about where we spent too much time on the slide, but I'll move on past it. Can y'all see this?
Yes.
Okay, I'm going to back out of this real quick to show you something else. Let's see. Remember me showing y'all this?
Yeah.
Mm-hmm.
Okay, and the one that's arched over is a club moss, and it's a diploid entity, and this fern is a diploid entity, but this bryophyte is haploid. Well, last weekend we were on that field trip with Rich Reeves, and I found some club mosses with the clubs on them, and the clubs are the top parts. That's where those are sporophylls. That's where the leaves are bearing spores. You can't see them, but they're different from the regular leaves. The one on the skinny one is the one, in fact, when they're both wet, they look close, very similar. The skinny one is, I hadn't seen before. And well, I saw it 2 weeks ago near my home, and then I saw it again last weekend. But it's Lycopodiella oppressa, and the leaves are oppressed to the stem so tightly, but you can see the cone part up above. The one, the big one here where my finger is actually the same as the one I showed you a moment ago, Lycopodiella alopecuroides. In fact, we found 4 club mosses on last weekend. That was quite astounding. But anyway, that's the club is dystrogulus, or cone where the spores are. And the other thing related to that is brings me to sporophyll, was one of the terms on the original matching. This is a sporophyll of a cycad, of female. The male has 2-foot-long cones, but on the female, the sporophylls look like leaves. And these big fat things on there are the seeds. So it's sort of a frondy looking thing, which but it's not a native cycad, Cycas revoluta. You might Google that and see what the male cones are like, quite a bit different, 2 feet long and weigh quite some pounds. But anyway, when you think about a fertile leaf, there's a good example of something that looks like a leaf. Now, the scales on a pine cone don't look like sporophylls, but they are. Okay. All right, and the petals on the flower may not look like, well, the petals have lost the spores of sporangia. But anyway, now let's pick up on this slide. And this is where I spent, whoops, what happened? I spent way too much time on this slide, but it was mainly trying to get you familiar with your own cycle before we tackle this one in the bottom left, and we'll not get to mention this one up in the upper right, the zygotic meiosis. It's typical of some of the primitive algae. I'm going to go ahead and try to minimize this. All right, now, okay, I've got to go through this business of getting my little spotlight. There it is. Now, all right, let me move this up out of the way best I can. All right, now I've added, I don't think, this thing at the bottom left, I don't think was on your original copy. Was it or not? I don't.
Not on mine.
Okay, it's not. Okay. Well, what I tried to do there was show you in reality that the sporophyte, this part up here is predominant and the gametophyte is less conspicuous, not cryptic, but close to it. Most of you have seen this. This is an alternation generations life cycle of fern. And most ferns are homosporous. That means they have one kind of spore. We'll talk about a few that are heterosporous. Lycopodium I showed you and its relatives are homosporous. They have one kind of spore in their cycle. But Selaginella is a relative of the club moss called spike moss, and it has two kinds of spores in the cycle, like higher plants. That doesn't mean it's an ancestor of higher plants, but you need heterospory, two kinds of spores to evolve into a seed plant. And these ferns don't have it, and the club Lycopodium doesn't have it. But since the sperm-bearing containers are called an antheridium, and the egg-containing egg containers called an archegonium are on the same thallus. Now, thallus is a 50-cent word, Thallus, is a 50-cent word for a non-vascular plant. Technically, a gametophyte, a moss a bryophyte is a thallus because it doesn't have any vascular tissue. Now, since does anybody remember in oaks what the term was when the male and female flowers are on different branches? How about monoecious? Okay, whereas willows or days are separate plants. That was the male and female flowers are on different branches, which are diploid. That's the term is monoecious. And I sent you an email that had a word called monicous, monoicous. Okay.
Oh, no.
Yeah, but that was some time ago. The point is an easier term to spell and remember is homothallic, same thallus, where the male and female sex cells are on the same thallus. If the sperm-containing ones and the egg-containing ones were on different gametophytes, we'd call it heterothallic or dioicous. Anyway, so sexuality here referring to the gametophytes is not the same spelling as monoecious, but monoicous. But anyway, you'd be hard-pressed. Probably couldn't even hardly find those. Go ahead. Don or somebody had a question. Maybe not. Anyway, I'll try to point that out again in a moment when we show you one of the slides that it's not in the original package I sent this morning, but I did send it a separate email. Anyway, what's going on here is the diploid predominates, and at this point, the gametophyte is dying because this sporophyte is becoming independent and eventually dies, and the sporophyte persists. And I've actually seen these, as I said in the, repeating myself from the first session, because I looked at it, these catapult-looking things are sporangia. I've actually seen them kick spores around. Each one of these little kidney bean-shaped structures is a sorus, which is a fancy term for a cluster of spores. People call them zits, Z-I-T-S. And they're about 5 or 6 millimeters in diameter, and they have a bunch of these in them, and they catapult the spores out. In ferns. Now, this is a slide I don't think you have at all. Well, I sent it to you, maybe it's an email by itself, but I think that's probably, that's the one that had the monoicous stuff in it. And it's not in the addendums slides. It was sent separately. But the point is here, and I wanted to, this is a fern, this is a moss. The difference, actually, all right, this cycle, unlike the one before it, you see the diploid predominates here, but in the bryophyte, the haploid predominates. And this diploid entity is this part right up here. Okay, it's a foot embedded in the gametophyte for food, getting nutrition, parasitizing it. The seta in the capsule where the spores are. The point, though, beyond that is, besides this being primarily haploid instead of diploid, is the fact that a lot of books will have a moth cycle and they'll have the archegonium containing gametophyte on a separate plant from the antheridium containing gametophyte. So, but this one, that would be heterothallic. This is homothallic or monoicous. In fact, the term unfortunately used here, it shouldn't be used, is monoecious. Whoever put this together, the book I got it from, did not understand the difference, because this term monoecious refers to diploid entities like an oak tree where you have male and female branches. But this term M-O-N-O-I-C-O-U-S or homothallic is when the gametangia or sex containers are on the same plant, different branches of the plant. Okay, and of course, if you're going to get the sperm to the egg, it pays to have them close, right? So something like the haircap moth, Polytrichum, has separate plants. You'd find that on one kind of plant and that on another kind of plant. But anyway, here's a young bud coming up. And if this fragments, that fragments, you can see where this would be an asexual form of reproduction in itself. So sexual and asexual at the same time. Let's see. 50% of moth species are heterothallic. That means not like this. And the other 43 would be like this. All right, now, but this has a lot to do with why bryophytes are not considered ancestral to higher plants, because higher plants have the diploid as predominant. On the other hand, it doesn't mean that the ferns are the ancestors of higher plants, because they're primarily diploid like higher plants. Now this is emphasizing, this is a generalized life cycle of alternation generations of which there's only one kind of spore inside of the sporangium, singular of sporangia. And there's diploid cells, spore mother cells or sporocytes. The last diploid cell of the diploid generation undergoes meiosis to form a tetrad, basically meaning four, myospores. And they're called, technically could be called myospores because they come from what? Meiosis. Now, there's a tendency to call it meiosis, but that's wrong. It's meiosis. In German, or in Latin, it's a diphthong, considered a diphthong. And then in this particular case, we got a gametophyte that has antheridium and archegonium on the same thallus, which is just like this. Now, and then producing sperm and eggs that technically, since we're going to talk about the three kinds of syngamy in a minute, but this would be fertilization or oogamy. Synonym for fertilization is oogamy, where the egg is large and non-motile, should be larger than that, but it's not showing here. Sperm is small and motile. Forming the first diploid cell becomes a zygote, and then the zygote forms an embryo just like in you it would, and then forms the adult or sporophyte. The difference is when you're mature, you don't produce spores, you produce gametes. Now, I jokingly call this the Holy Grail slide because number one, it actually has more information than you can grasp at one time, but it is a very important slide because it shows what happened here. Because it shows a comparison across the plant kingdom, actually even beyond the plant kingdom if you don't put algae in it. I showed you, let's go back and look at these two terms here. Let's go back to slide 34. Showed you this before, but Now is a better time, I think. Okay. Oh, what happened? Geez. It's a very sensitive thing here. All right. All right. Now, in this left one, it's a sperm container for a alga. Well, Steve knows what I'm going through, a very sensitive computer. Just look at it and it changes. But there's no jacket like in the bryophyte here or in the fern here around the sperm. And then the female would have an egg, its eggs would have no jacket around it in an alga with no protection. Kind of like, well, I joke about the bryophytes being like the amphibians of the plant world, but truthfully, the algae are the would be better in comparison, because the algae, the amphibians, most of them don't have containers for their sex cells. They just meet, in running bear meets little white dove in the water, so to speak. And that's true of algae. But in this case, these sperm cells come out when this breaks open and swim with raindrops and water down the neck of this neck that dissolves and then fertilizes that egg. And so it's still dependent on external water, unlike the higher plants. Okay. Bottom line, this is truly an antheridium. This is really an oogonium. And let's get back out of this slide and see if we can get back to around 260 or so, I think it is. Yeah, all right. Now, okay, so that alga had an oogonium that, and the male, unfortunately, is still called an antheridium, even though this one has a jacket and this one does not. So, all right. And they have one kind of spore in most ferns and practically all mosses. And then Lycopodium could be right here too, the club mosses, whereas a spike moss, Selaginella, another relative of Lycopodium, has two kind of spores in their cycle and has characteristics that higher plants have. Okay. These terms, this right here is probably one of the most difficult things to try to synonymize. That a megagametophyte is an 8-cell embryo sac. A microgametophyte is a 3-cell gametophyte. This male, this one's female. So megagametophyte, mega refers to female and micro refers to male. And they're inside of a diploid entity called the microsporangium. The pollen grain is inside of the anther and inside of the megasporangium or ovule is a megagametophyte. We're going to get into all that. And these are the modified leaves that support them. Here, there's only one kind of sporophyll because there's only one kind of spore. Here, we can separate, like I showed you a moment ago, that cycad. I was showing you a megasporophyll. So now, all right, this is a heterosporous kind of life cycle, like I said, and we have the sporophyte with two different sporangia with two different kind of spores producing separate gametophytes. Now, if you have two kind of spores, you're automatically going to be heterophalic, automatically going to be dioicous, because and the megaspore will form an archegonium containing entity, and the microgametophyte will form antheridia and produce sperm, whereas the archegonium will be housing an egg, and then they unite to have fertilization. And unfortunately, I didn't show this being motile, because this is very generic. But now the question I would ask is, what are these called on the previous slide? And the answer is the pollen grain and embryo sac. Back up. Remember, I underlined them? Okay. So the pollen grain is a synonym for the microgametophyte, and an embryo sac is a megagametophyte. And in the higher plants, it consists of 8 cells, one of which is the egg. This consists of 3 cells, 2 of which are sperm. Okay. We'll get to that, why not, that 2 business later. All right, this is an abstract this is the same type of cycle as the previous slide. It's just arranged differently. Instead of this being, all right, got haploid on top, diploid on bottom, they're turned 90 degrees so that we've got the gametophyte over here and sporophyte here. And I'm trying to emphasize here that the hallmarks of sexual reproduction are syngamy and meiosis. Everything revolves around those 2 important processes. This concept of differentiation is probably not an easy concept for you, but think of it as sort of a developmental morphogenesis or a developmental metamorphosis. Eventually, the haploid entity in meiospores will grow up and produce a thallus, and the thallus will differentiate sex containers, and having inside those sex containers, you'll have gametes that unite to form. This is oogamy because you got a small motile sperm and a large egg. Actually, there probably would be more than one flagellum here. Now, a human sperm has more like what you see here with the posterior sperm, whereas a lot of the plants will have anterior sperm. It's kind of like swimming with a breaststroke. And but anyway, that's a bit of a difference. But anyway, at maturity, instead of forming gametes like you would, the diploid entity or sporophyte produces 4 mother cells that start the process over. And this diagram is trying to indicate that in this overall here, this would be something we'll talk about toward the end of the day if we get to it, isomorphic alternation of generations, where the sporophyte and gametophyte would look potentially very, very similar, if not identical. Let's see what else I need to say. Yeah, just trying to indicate equal time here, more or less, spent in each generation. All right. Note the process. Okay, I already, okay, now that, yeah, I am going to dwell on the hallmarks in the next few slides of sexual reproduction, syngamy and meiosis. Let me get a swig of coffee. The one on the left is sort of a cartoon. One on the right is sort of an accurate depiction of the 3 kinds of syngamy. As you might expect, as you go from A to C, you get more highly evolved. Now, here's what a male gamete looks like among primitive plants, actually even higher plants. We tend to have this kind of thing, except for the angiosperms and one other group have no flagellae at all anymore. They just rely on the pollen tube to get them to that egg. But the human sperm is 1 flagellum and it's posterior, whereas this is anterior and swim like a breaststroke. Now, here's, let's see if I'll go ahead and do this now. I guess the best time. One of the very latest slides that I added this morning, the Chlamydomonas, which is an alga, has, at least the species I sent you, has this kind of syngamy. This has a female symbol, this has a male symbol. They are, and this is important, they're morphologically identical, but physiologically different, okay? Whereas these are morphologically and physiologically different, and these are morphologically different, at least. The female's bigger. We'll see at least one example of this today or next time in the form of sea lettuce, ulva, U-L-V-A. This is called isogamy, iso meaning the same. Anisogamy means not the same. And oogamy means 1 large non-motile egg and 1 small motile sperm. Now, question in the lab, in the lab, we could do this with algae, like Chlamydomonas, which is very tiny, or we could do it with a fungus, but we could put a Petri plate and put the female on one side. We call this minus, we call this plus, we could put plus cells on one side and plus cells on the other side. And when you do that, nothing happens. In another petri dish in the lab, we'd take minus or female type physiological, chemically different than that on the other side, on one side of the plate, and the same females on the other side. Again, nothing happens. But if you put the plus and the minus together in the same petri plate on opposite sides, running bear will meet little white doves, so to speak, in the middle of the petri dish instead of the middle of the raging river. And there'll be a line in the middle where they meet, and that is where they form zygotes. So the bottom line is they are physiologically different even though they look the same, even though they morphologically look alike. And so plus and minus will form the zygote in all three cases. Any questions? Now, again, this is primitive, less primitive, more evolved. Okay, now we're going to talk about, well, our goal is to talk about meiosis, but it's hard to understand meiosis unless you understand mitosis. Now, this mitosis is thought of as a sexual process. And what's the difference in mitosis and cell division? Mitosis, first of all, both mitosis and meiosis are strictly speaking nuclear divisions. And they are associated with the cytoplasmic division called cytokinesis, which means the cytoplasm goes along with it normally, goes along with the nuclear division. Now, Darwin said that evolution is descent with modification, but mitosis can be thought of as descent without modification, because over here you have exact duplicates of what you started with, okay? Exact copying of the cell. This is my favorite diagram of mitosis, but it has some problems. It comes from a lab manual back in the 70s when I was at Tennessee. Simple, but it shows some things and what the problems I'll talk about. But mitosis occurs in all cells that have a nucleus, vegetative as well as reproductive can undergo mitosis. Vegetative cells, non-sexual cells, as well as sexual cells can undergo mitosis. And again, it's usually associated with cytoplasmic division known as cytokinesis or cytoplasmic division, which is also true of meiosis. All right, now, what's wrong with the diagram? Chromosomes actually look like this during interphase when no cell division is going on, which is most of the time, most of the life cycle of the cell, 90% of the time, they're not dividing normally unless there are certain kinds of cells. But they don't look shortened, thickened, and contracted. In fact, if you looked at the chromosomes during interphase, inter means between, you would see chromatin dots or granules under a microscope if you tried to look at the DNA material. It's just kind of like, well, this pair, actually, now the question would be, mitosis starts here, okay, well, I thought I was asking that question here, but how many kinds of chromosomes are present? There's a pair of longs and a pair of shorts. So the N number is 2. N is, haploid means the number of kinds of chromosomes that there are, or pairs. There are 2 pairs. One would be paternal and one maternal. Okay, so so you could see that we wound up with the same number over here that we started with in mitosis. Meiosis, that won't happen. That will not happen. But the long ones is a homologous pair, and the short ones are considered a homologous pair. And that's a term that's used in genetics, which is our next topic. So it's not too early to think about that. But to better appreciate meiosis, to better appreciate what's going on in genetics, we look at this concept of meiosis. Now, what would happen if we didn't have meiosis in the human? Donna got a shot at that one. But what would happen is that we have 23 chromosome sperms meeting with a 23 chromosome egg, and that would make 46. But if we didn't have meiosis, the next cycle would be 92 and 184. So you can see the obvious advantage of meiosis. It brings half the number back down to the base haploid number, which is 23 in humans. All right, now, what is the haploid number here? The same as it was on the preceding slide. Haploid number, another way of asking it is how many pairs or how many kinds of chromosomes are there? There's still two, a pair of longs and a pair of shorts. The N number is 2, but the 2N number is twice that, okay? So 2N is 4. But notice in the end product over here, now, all right, this is confusing in a way. I didn't think about it till last night. Why are there two ways that it goes? And the answer is because if they sort with darks on the same side and lights on the same side, there's the option of doing the opposite. One light, one dark, one light, one dark. But they're still pairing up with one another, but they're pairing up in a different way. So there's two possible ways it can go, which is part of the reason the genetic variation can happen in meiosis. And, brothers and sisters being different and things like that. So just consider this one here and know that there's an alternative way of going. Now, all right, metaphase. All right, we'll stop here. Okay, and go down to, I'm not going to stop literally. I'm going to go down to, I was on slide 265. And if we're lucky today, and I don't think we'll be that lucky, we'll get to this slide, which is the same thing. It's the sort of the precursor to understanding genetics. All right, because what's happening is there's the male, the paternal or female. Anyway, we got one chromosome made of 2 strands that are called chromatids, and then lined up on the other side is the 2 strands from the other homolog. So these 2 strands could be from the paternal, from the mother. These 2 strands could be from the paternal or father, or the opposite. The point is, this is where the genetic phenomena that we call crossing over could take place. From here to here, there could be 1,000 genes. And so you get this intertwining going on, swapping of genes back and forth because the strands are crossing over with one another. That's not shown here. So what I'm trying to convey to you is, okay, let me back up here now. Obviously, I told you about the number significance of meiosis, but this has to do with the concepts. Besides the halving of the chromosome number, this meiosis at metaphase 1, which they're not close together like they were in the slide I just showed you, but they're touching, that's where you get a lot of genetic recombination. That's where you get a lot of differences. So meiosis is more important than just halving the number. It's a way to get variation in a sexually reproducing organism. Okay, so notice that over here, the haploid number never changes. The N number was 2, of 2 kinds or pairs. Well, there's no longer, there's only a piece of the pair, but there's still 2 kinds of chromosomes. So the N number stays the same. But this could be a spore or a gamete. It would be a gamete if it fused with another one, or it would be a spore if it grew into some haploid entity without fusion with another cell. Okay, now, This slide here doesn't, well, it's about all you get in some basic books because it's trying not to confuse everybody. To make sense out of this, it's perhaps better to start with something familiar. And most people say, oh, the most familiar thing to me is this flower. Well, I'm going to pull another one and say, the most familiar thing is not a zygote, let's face it, but we're going to start with that because you were once a zygote. Now, and so we're going to start with that. And the other hallmark of sexual reproduction is meiosis. And so a lot happens between these two. In fact, there's a fully developed egg inside, and this is a process. What happens is a lot happens between this and this, and that's why the next few slides are going to come in handy. A lot missing from this slide that we're going to see in the following diagrams. Now for how meiosis really looks like in a flowering plant. And it looks a little different than in the male and female, but look at this. See this foursome? Over here we got a foursome for the female, and here we have a foursome for the male. Let's back up. See this? That's what we got right there, the foursome. Mm-hmm. Tetrad. It actually even says tetrad. This is a tetrad of microspores. Now, ironically, in the human female, only one egg survives, one product of meiosis survives, and the other three wither away in the human female. Well, it happens in plants too, in higher plants. The three products of meiosis closest to the opening where the sperm come in, the micropyle, die out, and the one remains. That is the surviving functional megaspore. Now, we'll see more detail on that. As usual, females are more complicated than silly males. And that's true here too. And it's true. I don't mean that to be a sexist remark. It's just true. But anyway, all right, now each of the four microspores does what is going on here. And we'll talk about that in the very next slide. But look at the top of this, development of the male gametophyte, pollen grain, and over here it says development of the female gametophyte or embryo sac. Ironically, that's what's going on down here, because a mature male gametophyte has, it shows two cells, but the generative cell forms two sperms. So at maturity, how many cells does a male gametophyte have? Microgametophyte have? 3. But over here at maturity, a mature megagametophyte has 8. 3 antipodals up here that wither away, 2 polar nuclei that involved in higher plants in double fertilization, and then an egg in the center that gets fertilized, and 2 guide cells called synergids that also wither away. Total 5, 6, 7, 8 cells. So a fully developed megagametophyte has 8 cells, and a fully developed microgametophyte has 3. Now, and that's what the actual embryo sac looks like in a something like a lily. And the micropyle here is where the sperm enters. And I told you that in a bean plant, you can see the micropyle with your naked eye if you know what you're looking at. All right. Wow. That's a fancy word for the megaspore and microspore mother cell. Another name for the spore site is the spore mother cell, whether it be microspore site or megaspore site. Now, for the next slide, we'll go into the simpler, the male. So what you're seeing from here to here is what we're going to look at in the next slide. The anther of a stamen is where all this is going on, and that's a microsporangium. And we're going to have microsporogenesis and then followed by microgametogenesis, generation of the spores, microspores, generation of gametes. So the microspore mother cell, like here, the microspore site, undergoes meiosis to form the tetrad of microspores. Meiosis, there's halfway through meiosis, and there's halfway through the other halfway through meiosis. Now, I want to go back and point out something here that I should have pointed out. Now, don't write this down. You can't possibly write it down because it's going to be too fast. I told you mitosis is a simple replication of the parent cell. Well, meiosis is two successive nuclear divisions in which the first is a separation of the homologs. The second is a mitotic division because you get separation of the chromatids. And that's what's going on here. The separation of the homologs, separation of chromatids, if you could see the chromosome, but you can't. So the tetrad of microspores, from here on is microsporogenesis because what's happening from here on is what happens to all four of them. Microspore germinates to form a tube nucleus and a generative nucleus. The generative nucleus generates two sperm. The tube grows down the neck of or in the megasporangium of a gymnosperm or down the neck of the ovary of the style in a female, like a corn silk. You know, a corn silk is actually showing what's inside of a corn silk would be the sperm eventually. But actually, technically, the silk itself, I think, would actually be the neck of the style of female, and the pollen tube would actually be inside of it. You really can't see the pollen tube growing in a corn silk, but pretty sure the corn silk itself is the equivalent of a very long style. But anyhow, that the pollen tube grows down. And so at maturity, how many cells does a microgametophyte consist of? A microgametophyte at maturity is a germinated pollen grain, which goes back to this, way back to that Holy Grail slide here. Microgametophyte polygrain. Male gametophyte, same thing. All right. Now I'm jumping around, but maybe it's useful. Okay. All right. Bless you, I think, John. I think it sounds like Donna. Anyhow, all right. So, all right. Now the female is a little more complicated. We're going to go into it now. This is the best diagram I've ever seen to illustrate what's going on in megasporogenesis and megagametogenesis. We've got diploid tissue that's sporogenous in the ovule. An ovule becomes a seed after fertilization. Now, and these integuments are the seed coats that, like of the red skin on a Spanish peanut, that's what you're looking at here. All right, so the megaspore mother cell is the last cell of the diploid generation. And the megasporocyte or megaspore mother cell that's in a megasporangial wall divides halfway through meiosis and then fully through meiosis. And at the end, the three cells closest to the micropyle die, and the one furthest, the functional megaspore, remains, is retained. It's kind of like in the human female, it would be a lot of nutrition involved to bring all four to maturity. But at the expense of the other three, one of them is going to survive. The one furthest away from the opening of the micropyle where the sperm will enter. And then megasporangial wall is still showing, and the haploid cells inside are still showing with the three, withering away megaspores dying out. All right, from this point on, the second row is all about megagametogenesis. We've done the generation of the megaspores, and that megaspore starts to germinate and divide. So there's one mitotic division, there's two mitotic divisions, 2 cube, 3 mitotic divisions. 2 cube is what? 8. I'm terrible at math, but I know that much. 2 cube is 8. And so you got 8 entities, 8 cells. And the only thing happens between this and this is they migrate to different parts. The egg migrates to where near the micropyle, and the rest of the cells, the antipodals, are down here. And the fusion nuclei, also known as polar nuclei, is where that second sperm is going to, this other sperm is going to fertilize those and make in angiosperms, it's going to make a primary endosperm nucleus. And the gymnosperms, this other sperm is just going to die. It's not going to do anything. So we'll get into that maybe next time, if not today. In fact, I need to look at what, okay, now. So development of the embryo sac. This is the embryo sac. And funiculus is just a fancy word for the stalk. It's kind of like an umbilical cord that hooks into the placenta. And imagine a bunch of these hooking into what we saw in a cucumber or the luffa sponge from some time back I showed you. Okay, this is kind of the details of what's going on in here at double fertilization. We'd already said that, let's see. All right. This is best way to characterize is double fertilization. The one sperm fertilizes the egg to make the zygote. The other sperm unites with the two polar nuclei to form a primary endosperm nucleus. And the triploid entity, or sometimes higher than that, triploid is more nutritious than what you find in gymnosperms, because gymnosperms, the zygote and embryo are going to feed on the old haploid tissue. And this is a predetermined food source. It's richer in nutrition than the haploid thing that gymnosperms have. Gymnosperms do not have double fertilization because the other sperm just dies. And so the embryo feeds, and we'll see that later today, if not next time. But okay, now this is a good slide in a lot of ways, but it's got a few problems. The concepts gametophyte and sporophyte, haploid and diploid respectively, or what I'm trying to convey here. Most are familiar with sporophytes, and for mosses, the gametophytes. That's what you have, sporophyte of a moss, the gametophyte of a moss. Fern, there's the gametophyte that's about as big as your thumbnail, and the Antheridia are down here, and the Archegonia is up here. And this is the diploid frond that you're familiar with that I showed you earlier, like in that Riker mount. And the problem with this, if there's any, is that the cone is not haploid. Everything, in fact, what's inside of this seed of a pine cone, and there's two on each scale, what's inside is haploid, and this female gametophyte is haploid. These are the Archegonia, or what's left of the Archegonia. There's not much of a neck in the conifers. It's not your typical vase shape structure anymore, but functionally it's an Archegonium. This megasporangial wall down here with the integuments around it. Okay, in pine it takes a year between pollination and fertilization. Pollination was when that pollen grain lands and falls down in here, and it takes a year to grow through this megasporangial wall to get fertilization. And then in some gymnosperms it's quicker, but in pine it takes 2 years. This is a 2-year-old cone by the time it gets woody. When it's first year, it's maybe a ½ inch long, depending on the species. But in angiosperms, this is the most evolved of all. You have the sporophylls that have nothing on them, the petals. And it's hard to believe that the anther is a modified sporophyll, or the stamen is modified sporophyll, a microsporophyll. And the ovary that you can't see very well here is a megasporophyll. But the gametophyte is increasingly more cryptic as we go through evolution. I mean, it's not cryptic in a bryophyte, you can see it. Now, this diagram is a lot similar to this diagram, except this one has no human involved. The concept's the same though. In evolution, which we don't have a separate segment of here in this class that much, it's not as much as genetics, but we'll get into a little bit of it. But and I've added these circles, and I told you in an email to add these circles to yours because this is what you're familiar with. This is the one you're least familiar with, which we might get to today. But the algae, simple ones, but this is the alternation generations, like it's everything in this green area. In fact, that indicates vascular plants. This indicates embryophytes. Okay, everything in green is an embryophyte, which means that there's some protection from drying out of those sex cells, and that's having to do with an antheridium and archegonium. But as you go through evolution, the diploid entity becomes more dominant, the haploid entity becomes more cryptic, more hidden. Okay. In fact, even though they look different in size, this next slide is we're gonna go into the male, which is what we're doing now. All right. Technically on the right, the haploid female gametophyte is surrounded by diploid tissue. Okay, here we go. Let's label some of this. This line here could have megasporangium next to it because that's what's going on here. And out here, this is part of the integuments or seed coats that protect. And inside of, actually that's diploid, integument is, the megasporangial wall is diploid, but everything inside here that's labeled megagametophyte is haploid. And one of those sperms that's in pine, and you can tell pine because it looks like upside down Mickey Mouse. One of those sperms just disintegrates, but the other sperm goes through this megasporangial wall and eventually reaches this little pool inside, and there's an opening that they go in to fertilize the egg. So this is an egg here and an egg here. I actually show an embryo already, meaning the fertilization has taken place. But this is everything here is a megagametophyte surrounding these archegonia with no necks anymore. And then you get fertilization, which has happened here, which is why they can call this an embryo. And the wing is cut off. The wing is just like on this. This is the wing is cut off. So that's the same as this. All right, now, so the germinated pollen grain finds its way, and it takes a year to go through here on the cycle on pine. Cycads and ginkgo have motile sperm that swim all the way to the egg without a tube. Whereas in conifers and a group you probably don't know called the gnetophytes, Gnetophytes, have sperm with no flagellae that are conveyed all the way to the egg by means of a what? Pollen tube. All right, now in pine, all right, this is what we're trying to do is visualize what's going on in pine. Pine is actually it's polycotyledonous, unlike dicots. You can see that two cotyledons, and you can see part of a third that would be in front of and behind the slide. So unlike, in fact, here's the polycotyledons here. These are all many, monocots have one cotyledon, dicots have two, but pines have many, or conifers, gymnosperms tend to have more. And that's what you see going on here. But the important part of this is, all right, what identify the structure demarcated by the bracket in this longitudinal section of a pine seed? What's exploiting? That's the embryo. It's diploid. There's where the young stem is going to be, and the root is here in a pine seedling. And inside the seed is the old megagametophyte tissue that the ploidy tissue around the embryo. What's its function is the mother, the old maternal tissue that the embryo is deriving nutrition from. And there's no endosperm here. This is any nutrition, I guess, even though it's haploid, it's better than no nutrition. So this embryo, is unlike an angiosperm, is not getting high nutritive, high ploidy tissue nutrition, but it's feeding off the old maternal tissue. So that's what's going on there. This slide I've already showed you shows the increasing cryptic aspect of the haploid generation and the prevailing diploid generation becoming more conspicuous. It's just, this is microscopic. This is microscopic. This is very visible, barely visible, and increasingly visible gametophytes as you go back in evolutionary time. This is a summary of the heterozosterous life cycle in a seed plant, higher plant. And let's see. Okay. Yeah. These just the details. The leaf that bears the sporangium is called the microsporophyll in the case of the male and the megasporophyll in the case of female. If this was homosporous, we'd just be talking about a sporophyll, like a bryophyte or most ferns. But anyway, this is just another way of looking at what we've been looking at the previous few slides. Now, this is where the addendum starts. We got a few minutes here. I'm going to try to start it. There's actually 6 slides here instead of the original 3 that I sent you. And some of them I sent you this morning. But all right, let's see. All right, really the question comes down to isomorphic and heteromorphic alternation generations. Everything we've looked at so far has been heteromorphic, and you can tell them apart pretty easily. But in an isomorphic generation, alternation of generations, that's not the case. And as I say here, hardly anybody would talk about this today in a college class because it's just our educational system is dumbed down. And everything has been, like I said, heteromorphic, but now we're going to look at isomorphic. In a simple alga like Cladophora, these gametophytes look exactly like a sporophyte, superficially at least. But a sporophyte, as by name you think, produces what? Spores, and it does it by the meiosis of the spore mother cells. These cells differentiate in this filament and are diploid, and they're the last diploid cell that there is. And it forms haploid spores that unite. Don't unite. Spores don't unite. They grow into adults. And then this one has gametes on it, and this one has gametes on it. This filament, this thallus looks superficially like that, and that thallus looks superficially like that. So the sporophyte and gametophyte are what? Iso means what? The same, and they look identical, and that the hallmarks are still here. And frankly, this half and half business is a better way of looking at it because it's pretty much the case. This sporophyte is not prevalent in the cycle over these, not to my knowledge. Now, I'm going to throw in this little caveat that the filaments can fragment, both of them, so you can get asexual reproduction going on at the same time as you get sexual reproduction going on. No animal can do that. Even this simple alga, it's got work circles around animals when it comes to reproduction. Now, the difference is the thallus is not a filament. The thallus is two cells thick. This thallus is membranous and 2 cells thick. And the up above here at the top, we have a diploid entity called the sporophyte. And frankly, you had a little quiz question that looked more like this example, I think, than that. But the point is, all right, so the spore mother cells or sporocytes are inside of this, just like what's going on right here. These spore mother cells, except this is a filament. All right, this is a isomorphic alternation of generations. Instead of being a filament, this sea lettuce, a marine alga, has spore mother cells that form myospores that have 4 flagella instead of 2. They grow into, I don't like the terms male and female gametophytes or individuals, but that's kind of okay because what happens is there's a slight difference in the size of the cell, of the gamete. And what kind of syngamy was that this morning when we brought about it, when we introduced it? Not isogamy, because they're different. Anisogamy is what's going on here. It's not oogamy because they're both modal, but there's a slight difference in the size of the sex cells. And I think ova actually has all 3 types. I can't remember for sure. It has isogamy, oogamy, and anisogamy, but at least this species has anisogamy going on. But that the important thing is this is an isomorphic generation. If you're standing at the beach looking at these and this, you could not macroscopically tell them apart. And okay, and so spore mother cells are inside of here. It's the same thing, just illustrated a little more differently, showing an embryo, showing some things that were missing in this one. There's no embryo showing. Here, there's the embryo. It's also turned 90 degrees different. See, the haploid, diploid. Now, this is, okay, this is a heteromorphic alternation generations. The reason I'm showing you it is because it's very much like a higher plant in the fact that the diploid predominates. And in fact, what's different about sea lettuce, first of all, turn this 90 degrees. You'd have to, yeah, and you'd see the diploid here and haploid here, which is 90 degrees different from that. And in a kelp, how many, I'm not gonna, you can't really, off the coast of California, you might have a several hundred feet long kelp frond held to the substrate by a holdfast. And this is rivals, I mean, 300 feet long is not out of the question, which is rivaling a sequoia or some of the big eucalyptuses in Australia. The point is the diploid entity in this alga is huge, but the haploid entity is cryptic, microscopic. And so the cycle here is prevalently or primarily what? Diploid. And the haploid entity is cryptic and just little filaments and forming a zygote that forms an embryo, young sporophyte that forms a huge kelp plant. And there's some limited kind of vascular tissue, but it's fortunately in the marine environment, you don't need to have too good a vascular tissue because you've got water all around you, even though it's salt water. This is a generalized version of what you just saw, except it turned 90 degrees. And it's a repeat of slide 259 that we saw when we was talking about homosporous life cycles. So that's essentially what's going on here, except the diploid is predominant here and it's not equal time. All right, and then we'll go through this slide and then we'll quit because it's right at an hour now. This is the slide that I sent you this morning that I wasn't sure that you had. This is primarily haploid. And in other words, it's total opposite from you. The only diploid entity in the whole cycle is the zygote, and that's here. And that's what's going on here. Sometimes it's called zygospore because it persists for a while instead of a zygote. Zygospore, zygote, same thing. Just zygospore stick-walled. Now, to make, I want you for the moment, the next moment here, to forget about this asexual cycle and think about this sexual cycle, which is going on here. The difference, this one's going counterclockwise. This diagram that we've been showing you all morning is going clockwise. And I'm going to turn it 180 degrees. So now this is going that direction, and this is going that direction. And the gametes unite here at syngamy to make that a what? A zygote, which is here. And at meiosis, which is here, so put S here for syngamy, M here for what? Meiosis. And that's what's going on here. So you got haploid cells coming out of this zygote. They're called spores because they grow into an adult without fusion with another cell, which is the definition of a what? Spore. A spore grows into an adult without fusion with another cell. Just so happens the adult is just a bigger version of this. It's not a filament like we've been seeing. In other words, this is a filament, okay? And this is a excuse me, that filament is a thallus, and this filament is a thallus. In other words, this filament consists of several cells. That thallus consists of several cells, whereas this thallus consists of only one cell, but it's still a thallus, Thallus, a non-vascular plant body or precursor to plants, an oval cell. All right, now, so the only difference is maturity and differentiation of this adult cell. Undergoes a few mitotic divisions. It can't undergo meiotic divisions because it's already haploid. So it's undergoing mitotic maybe 3 or 4 times, and you get this capsule that breaks open, a container, a sex container that breaks open and forms gametes. And in this case, they're jelly or interlocked in the plus and minus. This is, by the way, Chlamydomonas, the one I was telling you about in the Petri dish. And the plus and minus come together to make a line of zygotes. The zygotes undergo meiosis to form these spores. Now let's look at this over here on the right. This is asexual reproduction. Let's just say, in this case, these were gametes. How do you know they were gametes? Well, let me ask you this. Well, asexual reproduction is carbon copies. So at this point, the only difference in that entity and that entity is the numbers. There happens to be 4 of them. There happens to be more than 4 of them, but fundamentally they're the same. They're both haploid because these are zoomeiospores. Now, and this adult produces something here and produces something here. The answer is what happens to them, okay? These fuse, that means they're gametes. These grow into adults without fusing another cell. So they're what? Spores. Okay, now, so what I'm driving at, what my final time here question to you right now is, all right, other than size, how would you tell zoomeiospore from something that comes from mitosis over here, from a zoomeiospore, from a gamete? How would you tell them apart if you were looking under a microscope? And the answer is by their origin and by their fate. This is heavy stuff. By their origin and by their fate. Now, since these spores are motile, we call them zo. Since these spores are motile, we call them zo, but they're different. These come from their origin is what? Meiosis. The origin of these zoospores is mitosis, so we could technically call them what? Mitospores. So what? Okay, so you can tell zoomeiospores from zoomeiospores by their origin. Meiospores come from meiosis, zoomeiospores come from mitosis. Now, how can you tell these from these? Well, unfortunately, we don't have a picture here to show when they're not locked together. But fundamentally, this looks just like this, which looks just like this, which looks just like this. The way you tell the spores from the gametes is by their fate. Gametes either die or fuse, right? Spores grow into what? Adults without fusing another cell. All right, we'll call it quits at that point. And this is a precursor to genetics anyway. The last few slides, let's see how many of them there are. Let's see. All right, 1, 2, 3, 4, 5. There's actually 4 slides leading into genetics, and that's by design. We won't meet next week because I'll be on the road home. But the 1st of August is when my next available time would be, and we'll see how it goes. And at that point, I'll have, by that time, I will have sent you the next increment 10, which will be genetics. And we won't probably finish genetics, but we'll get started on it. So, all right, we're going to back out of here and ask if there's any questions. Maybe I better stop sharing the screen. Uh-oh. It's not letting me do this. What's going on? Meeting controls. I don't know. I've never had this happen before. Jeez. Having a glitch of some kind. Return to meeting. Maybe that'll work. Not let me do that. All right, now.
If you hover over the bottom of the screen, will it toggle the control, the screen sharing control?
I would have hoped so, Steve, but now this is coming up. Let's see. No, it's not letting me do that. Wait a minute. Meeting controls. This is bizarre. Let me get rid of the annotation tools if I can. Right now, meeting controls. All right. You are sure to stop. Okay, stop share. Okay, good. Finally, geez. Sorry about that. I'm still not quite sure what happened there. Okay, are there any questions? I've done the best I could do, better than ever was done to me, but at the same time, that's still a lot. I hope I made some sense in comparing your cycles with other cycles and really biology in general when it comes to sexual reproduction. A little bit left should those 4 slides or so lead right into genetics. Is there any question that's burning that might help others if you ask it even? Okay, now so Kevin's gonna get to do all this by himself, I hope, but it all would have went better maybe if you did that 30-minute review of what I said. It's been so long. All right, I hope I haven't overwhelmed you too much, but there was a lot to digest there. It's a lot of terms, and that's why I tried not to do it all at one time. Okay, Amy gave me a thumbs up. So, all right. Well, y'all have a good rest of the weekend. And it's 80 degrees up here at the high today in Chicago, so we're lucky. Even South Georgia is lucky compared to the Northwest. So never thought I'd see that. But all right. Thank y'all.
Thanks, Bobby.
Huh?
Thank you.
Thanks. All right. You're welcome. You're welcome. All right. If you got any questions separately, you can email me.